The fact that we have an ability to produce any type of monoclonal or polyclonal antibody in a laboratory setting is actually very useful. And that's because it gives us a way to test for the presence of a protein inside a given sample. And that sample, for example, could be a blood sample or it could be a urine sample coming from an individual. So one important type of method that utilizes these monoclonal and polyclonal antibodies is ELIZA. And ELIZA is not only used in biochemistry, it is also used in medicine. So ELIZA stands for Enzyme-Linked Immunosorbent Assay. And this is a method that utilizes these monoclonal or sometimes polyclonal antibodies to determine and quantify the presence of some type of protein in our sample. Now, we're going to discuss two types of ELIZA methods in this lecture. We're going to examine indirect and sandwich ELIZA. And although these two methods are slightly different, they use the same exact concept. They use the same exact principle. So let's begin by examining indirect ELIZA. So the entire point of indirect ELIZA is to basically test for the presence of some specific type of antibody. So let's discuss the HIV agent. So let's suppose we have an individual who contracts HIV. And what that means is in their blood sample, in their blood, they're going to contain the antibodies against the antigen found in the HIV agent. So we can use indirect ELIZA, for example, to test for the presence of the antibodies against that HIV agent. And that's exactly what we're going to use to demonstrate how this process actually works. So let's suppose we have a well, a container, and inside that well, so on the surface of that well, we essentially place the antigens that are found in the HIV agent. So the surface of the well is coated with a specific antigen that comes from that viral agent, and these antigens are shown in red. Now, once we coat the well, we then extract the blood sample. We extract the antibodies from the blood from that individual, and we take these antibodies and we place them into this well. Now, some of these antibodies will not be against the antigen, and so they will not bind to that antigen. But in the case of the person who contains HIV, because they do have those antibodies against the agent, they will bind onto the antigen shown in diagram B. So these blue antibodies present in the blood of that person that contains HIV will bind onto the red antigens. Now, the next question is, how do we actually visualize the presence of this antibody-antigen complex? Well, we have to use a special antibody that we produced in the laboratory setting, and this antibody is manufactured in a way to basically contain an enzyme. So we have an antibody shown in green, and that antibody is created so that it contains an enzyme attached to it. And we'll see why that's important in just a moment. That's why we call this method enzyme-linked immunosorbent assay. Enzyme-linked means we create this monoclonal antibody shown in green that can bind specifically to this blue antibody, and that green antibody also contains an enzyme that will become important in step D. So in step C, an enzyme-linked antibody that can bind to that antibody of interest, the blue one, is added to our mixture, to our well. Now, if the antibody of interest, if that blue antibody is bound onto that red antigen, then our enzyme-linked antibody will bind onto this antibody as shown in that diagram. Now, in the next step, we basically want to first remove all the different types of molecules inside that sample that are not bound to anything. And so once we wash our sample, we remove anything that is not bound. And once we wash, we then add that specific substrate, that specific molecule that is converted by this enzyme that is linked to our green antibody. And once we add our substrate, that enzyme will begin to transform that substrate to some type of product. And when that transformation takes place, that causes a color change in our sample. And that tells us that the antibody is present inside that sample. Because if the antibody was not present in our sample, then following that washing process, we basically would remove any antibody that was not bound. And if that blue enzyme, if that blue antibody was not bound to our red antigen, then we essentially remove everything that was present in our mixture, including these green antibodies that contain the enzyme. And so following the washing process, if we add the substrate, because no enzyme is present in that mixture, the color change would not actually take place. So the color change signifies the presence of that antibody that we are actually searching for in the first place. Now, the darker that color change is, the higher the concentration of that antibody inside our mixture. So this method not only allows us to actually determine the presence of the antibody, it also allows us to quantify, to determine what the concentration is of that particular antibody in our mixture. So the entire point of this indirect ELIZA method is to basically test for that presence of our antibody inside that sample. For example, inside that blood sample. Now, if the individual did not have HIV, they would not have these blue antibodies. And so if these blue antibodies were not present in our mixture, the green antibodies would not have anything to bind to. And so once we wash our well, those green antibodies would be removed. And so once we add the substrate would have nothing to actually catalyze that reaction. And so no color change would actually take place. So this is how we actually use to test for HIV. This is what we use to test for HIV. Now, what about sandwich ELIZA? Well, sandwich ELIZA is very similar because it still uses this enzyme-linked molecule that we produce in a laboratory setting. But in the case of sandwich ELIZA, we test for a specific type of antigen and not for a specific type of antibody. So sandwich ELIZA is generally used to detect the presence of an antigen rather than an antibody, as we saw in indirect ELIZA. So let's take a look at the following 4 diagrams. So in diagram A of indirect ELIZA, we coat the surface of the well with an antigen, but in this particular case, we coat the surface with the antibody that can bind to that specific antigen that we are testing for. So the antibody specific to the antigen that we're testing for is attached to the bottom of the well. And then we take our sample, for example, the blood sample, or it could be a urine sample, and we place it into our mixture. And if the antigen is present in that mixture, it will bind to our antibody that is coated on the surface of our well. So we form the antibody-antigen complex. And now in step C, we add that enzyme-linked antibody that we synthesized in the laboratory setting, and we place it into our well. And if the antibody-antigen complex is present, if this mixture if this complex exists, then the green antibody that contains the enzyme E will essentially bind to that antigen. So this enzyme-linked antibody only binds to our antigen if it is bound itself to that blue antibody. And so if the antigen is present inside our mixture, we have this entire complex that is formed. And notice it's like a sandwich because we have 2 antibodies, and in between the antibody, we have our antigen. And so once again, as in step D for a sandwich ELIZA, we wash our mixture to basically remove anything that is not bound. And because this is bound, this is not removed. And when we add our substrate into the mixture, because the enzyme will be present inside our solution, that enzyme will begin to readily convert that substrate into that product. And when this transformation takes place, that once again causes a color change. So we see the principle that we use is this color change reaction. It's the color change reaction that signifies the presence of either that antibody or that antigen in our sample. So the well is washed to remove anything that is not bound, and the substrate is added to react with the enzyme producing that product that gives off that color. And just like in the case of indirect ELISA, in sandwich ELISA, the darker that color is, the higher the concentration of that antigen. And the lighter the color is, the smaller the concentration of our antigen. Now, if the antigen was not present in our mixture, then this enzyme-linked antibody would have nothing to bind to. And so when we wash it in step D, we wash away that enzyme. And so when we add our substrate, because the enzyme would not be present in our mixture, that substrate would not be catalyzed and we would not form that product. And so no color change would take place if the antigen was not present in our mixture.